Okay, right. So welcome back to this next video in which we are discussing the GQ pathway and the palmitoylation of IP3 receptors. Okay, right. So we are looking at the putative palmitoylation of the IP3 receptor subunits. Okay, and we now want to look at the enzyme complex which is claimed to be involved in the palmitoylation of these IP3 receptor subunits. Okay, and this enzyme complex is going to involve an example of a DHHC enzyme which will associate with another protein which is called selenoprotein K, and both of these proteins are within the endoplasmic reticulum. Okay, so let me show the endoplasmic reticulum here. And again, the endoplasmic reticulum has a lipid bilayer, so I will show this as a lipid bilayer rather than as a single line. Okay, so I'll draw two separate lines here to represent the inner and outer leaflet. Okay, right. And within the phospholipid bilayer here, we know that we have the IP3 receptor, which is this tetramer consisting of 4 separate IP3 receptor subunits. 1, 2, 3, 4, like so. Okay, now, the claim is that these IP3 receptor subunits are palmitoylated, and because palmitoyl groups are hydrophobic, this stabilizes the IP3 receptors in the phospholipid bilayer of the ER membrane. Okay, and if you remove these palmitoyl groups, which you can do by blocking the enzyme which puts them on, because they're not permanent, they will be gradually removed, so you have to continue to replace them. So if you stop the enzyme that puts them on, then the number of IP3 receptors that you have in the ER membrane goes down, basically. Okay, and this isn't because you have reduced the production of the IP3 receptors in any way, it's because their degradation, their removal from the ER membrane is accelerated, basically. And when you reduce the number of IP3 receptors which are in the ER membrane, then the amount of calcium that you will release by activating the GQ pathway from the ER is going to be hugely reduced. The calcium flux will go down, and this can have huge consequences for the signaling. Okay, right. So let's now have a look at the protein complex which is involved in adding these palmitoyl groups onto these IP3 receptor subunits. Okay, right. So to start with, one of the key players is a DHHC enzyme, basically. Okay, and specifically the DHHC enzyme that is involved in this is DHHC6. Okay, now let me show you the structure of DHHC6. So it's going to be in here. Okay, and I'll put it like so. Right, now I've forgotten to draw a very important domain, so I'll put it in now. Okay, and then I'll colour it in to hide my mistake. Okay, so I'll colour it in turquoise. No one will ever know now. Right, so let's say that this is the carboxylic acid terminus, and here is the amino terminus, and I've also forgotten to draw another very important domain over here. Okay, which again I will colour in a bright colour to cover up my mistake. Right, there we go. So, this is the membrane-spanning topology of the DHHC6 enzyme. So both its amino and its carboxy termini are in the cytoplasm, and it has these two important domains, one that is between the second and the third membrane-spanning alpha helix, and one that is after the fourth membrane-spanning alpha helix. Now, we'll start off with this domain here, this green domain. Okay, now this is what's known as the cysteine-rich domain. Okay, and for short, cysteine-rich domain is abbreviated to CRD. Okay, now, this cysteine-rich domain, as the name suggests, has some very important cysteines within it. And one of the incredibly important little motifs steps within this cysteine-rich domain. So remember, it will consist of a long length of amino acids, but there is a very important section of this cysteine-rich domain which consists of 4 amino acids, which are always conserved in all of the DHHC enzymes. So remember, there are 23 different DHHC enzymes. The one that's involved, or believed to be involved, in the palmitoylation of the IP3 receptor subunits is DHHC6. Okay, and this sequence that's within the cysteine-rich domain is conserved within all of them and is very important in the catalysis of the palmitoylation process. Okay, and this is the sequence D, H, HC, and this is why all of these enzymes are called DHHC enzymes. So, in order to understand this, you have to know your single letter amino acid codes. So D is the single letter amino acid code for aspartic acid, and I think I'll just show you the R groups of each one of these amino acids. So I'm not going to draw the full amino acid structure out, so I'm going to omit the core structures, I'm going to omit the amino group, I'm going to omit the alpha carbon and the carboxylic acid. I'm just going to draw you the R groups. Okay, so the R group of aspartic acid is basically a 2-carbon carboxylic acid side chain. Okay, so it's an acetic acid side chain like so. So that's the R group of aspartic acid. H, the single letter amino acid code H, stands for histidine. Okay, so the R group for histidine is a methylene group, like so. And then off this methylene group, you then have an imidazole ring. Okay, so this is a 5-membered ring where 3 of the members are carbons and 2 of the members are nitrogens, as I've shown. And you have a double bond here and a double bond here, and then you saturate everything else with hydrogen atoms. Okay, like so. So the name for this sort of aromatic ring structure here is to call it an imidazole ring. And the reason it's called that is that it contains this double bond between the carbon and the nitrogen atom. And basically, when you have a double bond between a carbon and a nitrogen atom, it's known as an imide bond. Okay, so that's where the imide comes from in imidazole ring. In addition, azo means pertaining to nitrogen, basically. So whenever you have something that has azo in its name, you can assume that there's something there to do with nitrogen, okay? And you've got 2 nitrogens, so dazole is kind of very nice, because that kind of suggests 2 nitrogens. So that's the logic in its name, that you've got an imide bond, you've then got 2 azos, dazole, and it's all kind of making sense. Okay, so you have 2 histidines, and then finally, the single letter amino acid code C stands for cysteine, and we've already seen the R group of cysteine. It consists of a methylene group with a thiol group sticking off it, so a sulfur and bound to a hydrogen atom. Okay, right. So that's this special domain here. Okay, and it's very involved in the catalyzing of the palmitoylation process. Now, what's this other special pink domain that I've highlighted over here? Well, this domain is what's known as a SH3 domain. Okay, and SH3 stands for Sark homology free domain. Okay, so Sark homology free, and some people put dashes between all of those. So Sark, remember, stands for steroid receptor coactivator, and it's homology free. Okay, so Sark homology free domain. Okay, so you have this special domain which is an SH3 domain, and that's just a structure within a protein that you find in loads of different proteins, basically, so it's a conserved domain. Okay, now, this SH3 domain is important because it's going to bind to another SH3 domain on another protein. So basically, in the ER membrane, you find complexes, okay, of DHHC6 with another protein, okay, here, which is selenoprotein K. Okay, so this is also a membrane protein. So this is selenoprotein, ooh dear, we've run out of space, selenoprotein K. And for short, selenoprotein K is often abbreviated to cell K, so CELK for short. Okay, and selenoprotein K also contains an SH3 domain, which I'll highlight here in pink on the cytoplasmic side, and this SH3 domain will bind to the SH3 domain of the DHHC6 enzyme, and they associate together. Okay, so you might be wondering, well, why is selenoprotein K called selenoprotein K? Well, the reason it's called selenoprotein K is that it contains in its active site a very special amino acid, and this very special amino acid is selenocysteine. Okay, so let me show you the structure of a selenocysteine amino acid residue. And again, I'll show this as a residue rather than as a pure amino acid. So I'll show it as though it's bound within a protein. After all, it is within a protein. Okay, so here is the core amino acid structure. The amino group, the alpha carbon, and the carboxylic acid group, both of the two end groups are involved in peptide links to the former and the after amino acid. Okay, and the R group then of selenocysteine is just like cysteine. So remember, the R group of cysteine is a methylene group and then a thiol group, but instead of having sulfur there, instead you have a selenium atom. Now, selenium is again in the same group of the periodic table as sulfur. So basically, in the periodic table, group 6, you have oxygen, then underneath it sulfur, and then underneath sulfur you have selenium. Okay, so selenium again has similar properties to sulfur and oxygen. So basically, you're going to have this selenium bound to hydrogen, and this is what's known as a selenocysteine amino acid. So the reason these are called selenoproteins, well, the reason there is a whole family of selenoproteins. This isn't the only protein which contains selenocysteine, but any protein that does contain selenocysteine is called a selenoprotein, and an example is this selenoprotein K, which is important here. So that's the logic in its name. Now, the claim of this paper is that firstly, selenoprotein K is associated with DHHC6 in the membrane of the ER. It is known that both DHHC6 and selenoprotein K are within the membrane of the ER. It's not known that they are associated into a complex. And also that selenoprotein K is utterly essential for the function of DHHC6, at least with regards to adding palmitoyl groups onto the IP3 receptors. Okay, so if you take the selenoprotein K out, and you can do that in two ways, either by knocking it out genetically, or by producing a dietary lack of selenium, basically. If you stop the selenium within the cell, if there's no selenium within the cell, then you can't produce selenocysteine residues. Okay, and that means that selenoprotein K production is halted. Okay, so those are two ways that you can knock out selenoprotein K. If you remove it from the ER membrane, then DHHC6 is no longer complexed with the selenoprotein K, and it doesn't seem to palmitoylate the IP3 receptor subunits anymore. So, the overall claim then is that DHHC6 is complexed with selenoprotein K in the ER membrane, and that this complex palmitoylates, adds palmitoyl groups onto the IP3 receptor subunits, so each of the 4 IP3 receptor subunits, and that these palmitoyl groups stabilize the IP3 receptors in the ER membrane and reduce the speed with which they are removed from the ER membrane. So, if you take those palmitoyl groups off, or if you bust this enzyme complex, potentially by knocking out selenoprotein K, then the palmitoyl groups will stop being added on, and the IP3 receptors will be removed from the ER membrane much quicker if they don't have the palmitoyl groups sticking off their sides. Okay, and this means that even though the amount that we're producing is the same, the actual number that will be in the ER membrane will be much lower because they'll last for much less time, basically. Okay, and that means that the amount of calcium you will release from the ER when you activate the GQ pathway will be hugely reduced, basically, and that can have dramatic effects.